Hi, welcome to my channel and let's solve these questions by topic today from coordination response and gene technology. So here the question number 1 says a number of insects can transmit diseases when they bite their hosts. The photograph shows Rhodnius prolixus, a blood-sucking insect that transmits Chagas disease between humans. This insect feeds on blood by extending its proboscis and pushing it through the skin of a person. It is thought to do this when it detects body warmth. So here's a picture of the insect, just magnified. Okay. The next part of the question reads, an investigation was carried out to study the effect of a heat stimulus on the proboscis extension response. Insects were kept in temperature-controlled container with a heat pad placed in front of them. The heat pad could be touched by the proboscis when fully extended. The heat pad was initially set at 25 degrees Celsius. It was then heated up to 35 degrees Celsius and the number of insects with a fully extended proboscis were counted. It was then returned to a temperature of 25 degrees Celsius. This procedure was repeated a number of times. So this whole is a procedure and an experiment. So what kind of questions do we often come up from the experiments and investigation? Let's see. The graph here shows the results of this investigation. Here is the graph. So question number 1A says, calculate the mean decrease in the number of insects showing PER each time the procedure was repeated. So what we have to do here essentially is we have to calculate the mean decrease in the number of insects showing PER each time this procedure was repeated. Okay, and in this investigation 350 insects were used. So one common question that may arise from an experimental question will be a graphical question. From here you may be asked to interpret data or you may be asked a mathematical question like calculating mean decrease or increase. So let us now see how to do this. Okay, so here I did the maths as you can see. Okay, let's break down what happened here in this math. So it's basic percentage drop math, and what happened is that at the end of the beginning of the trial, almost, if you see, almost 100% of the insect showed a repetition, right? Almost 100% of the insect showed repetition. Okay, so then what happened at the end? If you see, the line is touching just above, just at the zero. So repetition ended and show zero repetition. Okay, repetition ended. Okay, so we can say that across the experiment, the percentage of insects showing PER dropped from 100 to 0. Okay, meaning 100% drop in response. Now we have to count that how many times this procedure was repeated. So we took the base value first of all, that is 1. So there is one time the repetition occurred and then all the way of 58. The repetition 2 times for 58. Okay, and then we just minused 58 from 1 and then we got 57. So our repetition took place at 57 times. Okay, and then we worked out over here how much percentage dropped per repetition. Okay, so if our repetition occurred 57 times, so if we divide it by 100, we get the percentage drop of 1.75. Alright, as this happened across 57 repetitions. So basically, essentially, so every time the procedure was repeated, about 1.75% fewer insects showed PER. Okay, now that we have the percentage of fewer insect that showed PER, we can easily find out the mean decrease. We just write the percentage 1.75 divided by 100 and then we multiply it by 350 and we get an answer of 6. Are you following? I hope that's clear. Now let's move on to our part B of question 1. Alright, so part B asks us to explain the result of this investigation. So whatever is recorded in this data, all we have to do is explain the data in the graph. So what happened here is we have to write first of all that what this investigation or experiment is about. Alright, so we get the idea that from the question that it was about an investigation to carry out the study of the effect of a heat stimulus on the proboscis extension response. So it's an investigation to carry out the study of effect of heat stimulus on a particular species of insects. So we say whenever we are exposing a species to a certain kind of stimulus and we record their response, we say that this investigation demonstrates this investigation demonstrates habituation. We call this kind of experiment an experiment for habituation. Okay. Now, why do we call this experiment habituation? Because the response decreased each time a stimulus was repeated. Okay, and then we can, since it is for 3 marks and we have written only 2 points, we have to make sure to include another point so that we get the full 3 marks. And this point would be because they learned that the heat was not a source of blood. So let's sum this up again. So the basic information that guarantees 1 mark is if you write what is this investigation. This investigation demonstrates habituation. You get 1 mark for this. Done. Then we have to explain the investigation. We explain the investigation as this investigation was carried out because the response decreased each time a stimulus was repeated. That's the result of the investigation. Okay. And then we have to talk about, since it's 3 marks, we have to talk about why that particular thing or why that particular response happened. Or basically explain the result. Why did this result came? So because what happened was that the insects, they realized that the heat was actually not a source of blood. Okay, are we following? I hope this is clear to you. Now let's move on to the next part. Okay, the next part suggests why a drop in PER to a heat stimulus could be an advantage to this insects. Okay, so basically we know that uh, habituation is a response by animals, or if they are in a very unfamiliar situation. Okay, so in a way you can say that this heat stimulus that they are receiving, the animals or the insects in this case are receiving, that is due to not being exposed to this kind of environment or change in the environment before. Okay, so now they react accordingly to the change. Now, after they react to the change in the environment, in this case the heat stimulus, they will not continuously react. Okay, at some point these insects are going to stop reacting to that change because they simply become habituated. That simply becomes a habit for it. This is not causing them any harm. As long as it's not causing any harm, the insects are quiet to this change. Alright, so basically that's what habituation means. Now, our question exactly asked here suggests why a drop in PER to a heat stimulus could be an advantage to these insects. So, why does the insect decrease the response to the heat stimulus? And why is this an advantage to this insect? Is what basically this question asks us. So, we could simply write this as so that the energy is saved by not extending proboscis. Okay, and then we can write, since it's for 2 marks, we have to give another reason. So it's less likely to damage the proboscis. Alright, so this is our answer. So it's better if you can remember this point. So why does an animal become resistant to change in their environment, or why don't they show any more response to the change? So that the energy can be saved by these insects or by these animals, so that less damage is caused to them. Okay, that's our answer to part C. Let's move on to the next part. Okay. So in this question, we are given that multiple sclerosis is a disease that causes the destruction of the myelin sheaths around neurons. This destruction is caused by the person's own immune system. Lesions form in the brain where myelin is destroyed. The photograph shows lesions in the brain of a person with multiple sclerosis as seen using magnetic resonance imaging, also known as MRI. So the question asks us that calculate the actual length in millimeter of the lesion between point X and Y, whereas the actual length of the brain is 15 centimeter. Okay, so firstly let's understand what is this question really asking and what we are given. We are given an MRI image of the brain, as we can see. On the actual human brain, okay, the question gives us this data. The actual length of this brain is 15 centimeter. Okay, this is the actual human brain. The real size is 15 centimeter long. Okay, now if we measure the MRI, okay, wait, sorry, not good. Okay, so if we measure the MRI image with a ruler, okay, let's draw it up till here and here. Okay, please pardon my line, it's not straight at all. Okay, so up till here and up till here, we get an approximate around, if we measure it on scale, we get 63 millimeter, okay? Micrometer, okay? mm. And then we have to calculate the distance between these 2 points, point X and Y, okay? So this we get a point around, say, 6 mm on our scale if we measure it. Okay, now what does the question ask us? The question asks us that what is the actual diameter in mm of the lesion between X and Y. Okay, we have to understand and answer the actual diameter, okay, not the diameter given in the image but the actual one. Okay, so in simpler terms, the lesion appeared to be 6 millimeter, micrometer apart on the MRI image. So how far apart are they in real life in the actual brain? So that's what we have to find out in this question. Okay, so this is a magnification question, but instead of finding the magnification, we are using it to find the real size from the image. Okay. Now we have to take the scale factor instead of magnification. Okay, whenever you're given a question like this, use the scale factor. This formula will be actual size, I'm writing S instead of size, by image size, okay? Image S. Alright, so also one thing, let's convert this so that all the conversion, always remember all the conversion must be in one unit, in the same unit, okay? So all of here is micrometers, so we must convert this into micrometer as well. This gives us centimeter equals 150 mm. Alright, now we have converted all the units to the same. Now we can do this maths very easily. So our scale factor would be, oh god, there is so less space. Okay, just let me do it here. I hope you'll understand. We can write the answer here. So scale factor, I'm writing it as SF. We get the actual size. The actual length is, as we have calculated, 150, and our image size from here we have gotten it 63. We get a scale factor of 2.38, and this means that everything on the image is about 2.38 times smaller than in real life. Alright, so now we can use the scale factor to find the actual lesion size. Okay, so the lesions appear to be 6 mm apart on the image, right? So the actual distance between the x and y is basically 6 mm into 2.38, because everything we see on the MRI scan and everything in real life has a scale factor of and a difference of 2.38. So obviously we multiply it with the measurement we see on the MRI, which is 6 mm. So then we get an answer of 14.28 that we see from calculating it. Then if you want, you can keep the answer like this, totally alright, but it's better to round it off. Alright? Because always try to answer in 3 significant figures. So 14.3 mm is our answer. Here, very easy. Okay. So the next part of the question says the table shows the speed of conduction of an impulse along a non-myelinated and myelinated neurons with axons of different diameters. Okay, so here we have the two types of neuron, that is firstly non-myelinated and other is myelinated. And there is the diameter of the axon given in uh, micrometers, 1, 2, 3, 4, 5, 1, 2, okay. And then we are given the speed of conduction of impulse, okay. Okay, now what does the question say? Number 1, okay. 1 says show that the effect of diameter on the speed of conduction of an impulse is 6 times greater in a myelinated neuron than in a non-myelinated neuron. So basically we have to show that the effect of the diameter on the speed of conduction of an impulse, okay? So we know that if a neuron is myelinated, then the speed of conduction of an impulse would be greater due to myelination, and if it is a non-myelinated neuron, then the speed of conduction would be lesser, okay? So we have to show the effect in terms of the maths, okay? How we can do this math, I'm showing you right over here. Okay. Basically, let's just calculate the mean, okay? Myelinated mean increase. So we have to calculate the mean increase for mean increase in conduction per 1 micrometer increase in diameter for each type of neuron we have to calculate, okay? Just a second, okay. myelinated. For myelinated, we are given the mean increase. So to calculate mean increase, we have to do the math like this, okay? Minus 2.6. We divide this by 2, then we get 2.95. Alright? So this is the mean increase in conduction per 1 micrometer. Increase in diameter for each type of neuron. Alright, this is for myelinated neuron. Now we calculate it for the non-myelinated one. So for the non-myelinated, sorry, my writing is all over the place. Sorry for the clutter, please forgive me, but I hope you can see the step I'm doing clearly because that's more important. Okay, for this one you have to minus 4.2 minus 2.3 because of the 1% 1 micrometer increase, and then you have to divide it by 4 because we have taken these two values 4, then we get the value around 0.475. So basically now that you have gotten the individual values for the mean increase in both the type of neuron, now you could just divide this. You could just divide this by each other. So if you divide the myelinated by the non-myelinated, if you divide this both by this, okay, then we get 6.2 or 6.21 in this case. So that's our answer. That's your you'll get 2 marks for showing this one, and you'll get 2 marks for showing this one. All right. I could have done it literally here, but I've done that there. Okay, no problem. When you solve that, you guys make sure that you're doing it within this blank space. Okay. The next part of the question says, explain the effect of the myelin sheath and the diameter of the axon on the speed of conduction of an impulse. So here we have to explain the effect of the myelin sheath and the diameter of the axon on the speed of the conduction of an impulse. So basically we have to explain that why the speed of conduction of an impulse is greater or lower in terms of a myelin sheath. Okay, so if a myelin sheath is there, why the speed of conduction is greater, and what role does the diameter of the axon also plays in the speed of conduction of an impulse. So because basically all the reasoning stuff. Okay, so you have to structure your answers nicely. So let's do this. First of all, we have to talk about the effect of myelin sheath and diameter of the axon on the speed of speed of conduction of an impulse collectively. So we can write that both cause an increase in diameter and the presence and the presence of a heat increase speed of transmission, right? This is our first point that we're going to get marks for. Okay, both cause an increase in diameter and the presence of myelin sheath increase the speed of transmission. Okay, both cause an increased diameter and the presence of myelin sheath increases speed of transmission. Okay, this is our first part. So when we increase the diameter and when we increase the presence of myelin sheath around the neuron, this both can increase speed of transmission. Okay, please memorize these points because this kind of question comes often in the exams. Okay, now let's talk individually, like how myelin sheath affects the speed and how the diameter affects the speed. All right. So for myelin, we write myelin insulates the axon because it is hydrophobic. So myelin is hydrophobic, meaning that it is water hating, it doesn't mix with the water and thus insulates the axon. Okay, so this therefore nerve impulses jump from node to node. Okay, so since myelin insulates the axon because it is hydrophobic, the nerve impulses jump from node to node. Okay, if you have a problem in this particular question or in this particular topic, then I suggest you watch my playlist on topic 8A because this one is from topic 8. Okay, now we talk about the effect of diameter of the axon on the speed. And finally, diameter increases the resistance and as diameter increases the resistance to flow decreases. So what essentially happens here that when these impulses jump from node to node, their speed actually decreases because they're facing resistance from the other side. Alright, so what the diameter does, as it increases, the resistance against the flow also decreases. So the nerve impulses transmit more quickly. Okay, so that's your full reasoning. read this again, and as I emphasize the importance of these questions, because these are the common questions, you may come across this question more often. In a second, I'm explaining the whole answer again. Just a moment, please. Okay. Okay. So let's review a little bit about myelin sheath here. So we know that myelin sheath is an insulating layer around the neuron and axon, actually, around the axon, and it increases speed of transmission. It insulates the axon because it is hydrophobic, alright? And it allows saltatory conduction. Do you remember what saltatory conduction is? The electrical impulses that jumps from one node of ganglion to the next instead of traveling continuously. Okay, this greatly increases the speed of impulse conduction. Okay, and then let's talk about the effect of axon diameter. A wider axon means that there is less resistance to the flow of ions inside the axon. So basically, when the impulse is passing through the axon, there is a resistance to the flow of ions inside the axon. So this resistance decreases in a wider axon. Okay, this allows the action potential to travel faster along the axon. And also remember additional point: a larger diameter means the axon can carry a stronger electrical current. Okay, so simple. Remember these points, and if you face another question like this in the exam, you'll be good to go. Now let's move on to the next question. Okay, this is symptoms of MS. Okay, so do we remember what MS is? MS is basically the demyelination of the myelin around the neurons. So as we know that myelination basically results in faster impulse transmission in the axon, but when there are demyelination around the neurons, the opposite happens. Demyelination is in a person with MS results in slower impulse transmission. Okay, and then let's talk about the blindness in one eye and lack of coordination. So when we're talking about the brain specifically, our brain has two parts, right? Northern and southern hemisphere. And so we are referring here to one side of the brain. Okay, so that side of the brain is demyelinated. As a result, that side of the brain, or that one particular side, is affected more than the other side because this one is demyelinated. So this causes the blindness in one eye and the lack of coordination due to the demyelination of one side of the brain. Okay, not both, but just one part. And now we have to explain the symptoms and the effect of the lack of coordination. Okay, this one would be lack of coordination. Lack of coordination. Coordination will result from will result from sensory neuron sensory neurons and motor neurons transmitting nerve impulses at different speed. Okay, at different speeds. Sorry. I'm really sorry if you can't understand my handwriting. Please pardon me. Okay, and an additional point to be noted is okay, and this is another point. Okay, blindness in one eye will result if demyelination occurs in the part of the brain which is associated with vision or coordination. Like I said, that it occurs only in one part of the brain, and that part is the part basically which is associated with vision or coordination. All right, clear? So this was it for the video. We have covered several topics, important topics here today, about habituation and saltatory conduction and some very topic related question from our topic 8A. I hope you're clear. If you have any question, feel free to comment down below or ask me. And yeah, I hope to see you in my next video. Thank you so much for watching.